Welcome everyone. Thanks for joining. I'll introduce you to Max. Max is an assistant professor in the Department of Psychology at University of Minnesota now. He studies individual differences in cognitive and brain aging. He received his BS in psychology from Minnesota itself, and he spent 2 years working as a research assistant at National Institute of Mental Health in Bethesda, Maryland. Then he received a PhD in clinical psychology at Duke, where he worked with Ahmad Hariri, Terry Moffat, and Avshalom Caspi. He then completed his clinical internship at McLean Hospital, followed by a postdoctoral fellowship with Randy Buckner at Harvard University. And we were colleagues at Randy's lab, and I had great time discussing things with Max. And again, he has this wonderful ability to come from both sides, the clinical side of things as well as the mathematical and dense analysis side of things. He has this amazing paper on test retest reliability, which is the most cited paper on this particular topic. And he's doing amazing work, and probably talk a bit about aging today. And I think this paper got recently published, right? So
Yeah.
talk a bit about that. So again, over to you, Max.
Yeah. No, thank you for the invitation. It's fun. It's fun to this is my first talk across the world. It's so fun. I love this. So thank you for staying up late to join with me. And feel free to I mean it's we can kind of see how it goes, but feel free to jump in and ask questions or type a question in the chat, and Vaibhav can interrupt me and ask it. I'm happy for this to be a discussion as well, and I left plenty of time that we can talk afterwards as well. So I will share my screen and get started. Um, yes, can you all see that? Okay, second. Okay. Um, so yeah, I want to talk about this paper just got published over the summer. And so take a look. I'll talk through like some of the basics of the paper and have save some time to talk about where we're going next. But this is work that I did during my postdoc, and it's kind of a collaboration with my PhD lab and kind of integrating some of the ideas I developed as a postdoc as well. Um, and it's fun to see Vaibhav again. We were right down the hall from each other. He was like, maybe his desk was probably 15 feet from mine just a few months ago. It doesn't feel like that long ago, but it's crazy. Um, okay, so I'm going to talk about what to think about this idea of what is a brain clock. Some of you may have heard of this, and I'll kind of get into the weeds of it and talk about what we're doing to try to make what we think is a little bit of a better brain clock. Um, just to start off, I want to play a little game with you all. So maybe type in the chat or just shout out. These are two images of faces, and I just kind of want to just to get people's judgments. What if you had to guess and you have to put a number on it? How old is the face on the left? Shout out a number, anything.
35.
31. Okay, that's we got it. We got one. I think most people probably say something around that, maybe. Any other guesses? Okay. Oh, we got a bunch in the chat. 31, we got 40, mid 30s, 36, 45. Okay. So we're like between 30 and 40, 45. Okay. How about the face on the right? Let's do the same thing. Type in the chat or shout it out. How old do you think the person on the right is?
Maybe 40 plus.
So, yeah, okay, we got like 45, 50, 55. We got some 60. Um, okay. So I think just like a rough, quick and dirty estimate, I think we're talking about somewhere between like 35 and somewhere between 55 to 60. So we're maybe like thinking like this is a 15 to 20, maybe even a 25 difference in how old these two individuals are. And the reason I start with this is because I think this captures a really core feature of what I study in my research program, is that these people are these are actually not individual people. These are both like facial averages of 10 images. And in particular, all of these photos come from individuals who are 45 years old. So all of these people are from a study that I worked on as a grad student, which is every single person born in a single hospital on the South Island of New Zealand within 1 year. So every single image that went into these photos, the individuals were born within 1 year of each other. However, the 10 photos on the left were the 10 photos that were rated by independent raters as looking the youngest, and the 10 photos on the right were independently rated as looking the oldest. So what you can kind of see here is that our brains, our minds are like immediately picking up on something that's different between these photos, right? Like there's something about aging that is captured by someone's face. And I think this is all intuitive. We make these judgments all the time. You're walking down the street, you're in a coffee shop, you're immediately, without even thinking about it, making a judgment of, oh, this person's maybe my age, or this person's maybe my mom's age, or this person probably just graduated from high school. And this captures what I like to call biological aging, that individuals of the same age can actually be underneath biologically aging at very different rates. And I am very interested in these individual differences because I think it gives us some insight into what we can do to both understand the aging process better and also hopefully at some point have interventions to slow down the aging process to help prevent age-related disease later in life. This isn't just true of the face. It's not that we're just really good at judging how people's faces are different. This also happens in another organ that I care a lot about, which is the brain. So here's just a quick cartoon to highlight some differences between what you might see on a younger or older brain. In this image, this is like if we did an axial slice cut through the brain. So on the left-hand side, we see what might be a very young-looking brain with pretty small ventricles. They're really clean white matter, pretty thick cortex, and like you can imagine like if the skull was here, it's kind of filling out the whole intracranial cavity. On the right-hand side, we have what might be an older looking brain with like larger ventricles, little dark spots in the white matter that people call it dirty white matter. These might be called white matter hyperintensities on an MRI scan. And they have thinner cortex and reduced overall brain volume. So in the brain, we can have if you kind of look across the lifespan, there's all of these age-related trends of just what's happening as people are getting older on average. And this is similar to these faces. This happens among two people of the same age. They can be accelerated on this aging curve, right? They can be aging faster and have an older looking brain. And we can use this to do science. We can try to understand like what is it about someone's life that is causing their brain to age faster or slower. And we can look at someone's brain and say like that their brain looks older than we might expect for their age. And that idea is at the core of what we're going to call a brain clock, measuring how old someone's brain looks or how fast it's changing based on how their brain looks, measuring how old they are or how biologically old they are based on how their brain looks. So I'm going to use the term clock to refer to this just because it's kind of a convenient term that everyone can imagine. Your brain is kind of telling time. How old are you? So why are we interested in brain clocks? I'm sure some of you, if you've ever done MRI research or read MRI papers, you might have come across these. They're pretty popular. But there's also clocks in other kinds of research. There's blood-based biomarker clocks. There's clocks of just from like facial imaging. You can train an AI model to predict how old someone is. These are useful in a lot of ways. And one way that I like to think about these is that aging is the gradual, progressive, and correlated decline of multiple organ systems that accelerates the risk for many chronic disease. So fundamentally, like when we care about thinking about how fast someone is aging, we care about it because of this definition of aging. And importantly, people age at different rates. So there's pretty large individual differences, especially as you get later in the lifespan, that And this has consequences for someone's health outcomes, right? Having a stroke when you're 68 is very different than having a stroke when you're 94. If we can push these kind of age-related decline events later into the lifespan, we can live more years of healthy life. To study and slow aging, we need effective measures of how fast someone is aging. And I think this is pretty intuitive, but just to highlight it, like if we had an intervention that we thought could slow down how fast someone is aging, we would need to be able to measure biological aging at baseline, give some intervention, and then measure it again at follow-up. And we need to have really sensitive measures that captures the underlying process that we care about. And this is particularly interesting, I think, for the brain, because many of us care a lot more about brain aging than we do about other aging, because as soon as your brain is starting to decline, it really impacts your quality of life. It really impacts your ability to take care of yourself, live by yourself, work in a job, all of those kinds of things. So the gold standard of how to measure the rate of biological decline is to measure decline across multiple organ systems over time. And this is really key. So, like, aging itself is a longitudinal process, right? Each of us is aging. And in order to know how fast we're aging, we need to measure something that is changing over time. And hopefully, we measure it over a long period of time and we measure it quite precisely. But the dilemma of these kind of gold standard measurements of change over time across multiple systems is that they're typically very expensive. It's hard to measure multiple organ systems. And it's especially hard to measure them over time in large cohorts because you have to wait, right? Like you have to measure a bunch of biomarkers and then you have to wait a long time and you got to get all these people to come back into your study years later and you got to measure them all again. And that takes a long time and it's hard work. And if we want to accelerate our science so that we can measure these things faster and we can test more interventions and we can really understand aging better, we need to find ways of capturing this aging process faster and cheaper, ideally. So one possible solution are these aging clocks that we've started talking about, that if you could get a good proxy measure of how fast someone is aging or how kind of biologically old they look from a single time point, and we actually could convince ourselves that it does a decent job, that could accelerate our science and help us get these proxy measures for biological aging to do interventions, to do science faster. So the kind of core summary here is that developing biomarkers of aging is a major priority for gauging and intervening on risk for late life chronic disease. Another kind of feature of this that is just worth highlighting is that it's really hard to intervene on age outcomes once the problem is already like here. So once you have Alzheimer's disease, it's pretty hard to do anything about it. But there's lots of reasons to think it's a lot easier if we could have interventions earlier in the lifespan. Um, like for many reasons. One of them is just that like younger people can handle more intense interventions. Um, and if you can even make small differences in a young person's trajectory, and young here, we can even say like midlife, or even like in your 50s and 60s. But if you can do something earlier in the lifespan, it can have larger effects later on just by slowing it down a little bit before the problem is here. So being able to measure something across the lifespan that we could pull forward and measure earlier to see if we can have like prevention efforts is a major goal of what we would call like geroscience. So let's just talk through this a little bit further and feel free to jump in. Any questions or does this make sense so far to people? I have a small enough group that I think you could jump in if you want.
Question in the chat. How do we create a baseline if all individuals age at different rates? Are there common features of aged brains, and how accurately can we approximately age different organs?
Yeah. Yeah, I mean, I think this is really so you're getting at the key that I'm going to touch on this a bit more in the next few slides, but this is like these are really important questions. The kind of short version of the answer is that there are a lot of different ways to try to solve these problems, but I don't think there's one kind of widely accepted answer. Um, real advantage is that if we can, we don't have to care so much about baselines if what we're actually ultimately measuring are change within individuals. Because what you're doing then is you are your own self-referencing baseline. So I don't really care about where you started in one version of this kind of measurement. I don't really care about where you started. I just care about how fast you're changing of change within an individual. Um, we could talk about like health status or like overall risk. And in those cases, we really care about baseline. But if I just am trying to say, how fast are you aging right now? Or how fast are you aging over a small period of time? I just want really precise measures of change within individuals. And I think Vaibhav said that you guys have started diving into some of the precision fMRI work. And there's kind of a similar intuition here that like, one way to really dive deeper and develop better measures is to just care deeply about individuals and quantifying individuals' change. It's not exactly the same problem we're solving, but it's a similar intuition. But I'll touch on this more, and please jump in if there's more questions.
Hi Max, is this
Yeah.
a unidirectional thing or the changes can reverse also? Like you said, you want to just measure the rate of change or how much an individual changed. So you are assuming that the process is a unidirectional process or it can reverse also.
Yeah. I mean, in theory, it could reverse. And I mean, if we had like like true rejuvenation interventions, you could reverse aging. Um, one of the convenient things that you're getting at here, I think, is that one of the convenient things for our models is that a lot of age-related declines are just unidirectional in most people for the vast majority of the lifespan. Once you get past like, puberty and like adolescent developmental changes, a lot of things in the brain and in cognition are kind of going in one direction. That's not true in every case, and there's exceptions to this. But for the most part, your cortical thickness, I think, peaks at the age of like 3. Your brain volume peaks at the age of, depending on what you're looking at, maybe like 20. And a lot of these metrics, like how many white matter hyperintensities you have, those that like peaks, meaning it's at its lowest at birth essentially, and it's just a one directional change. That doesn't. We could imagine fitting models that are more complicated than that, but like for a lot of our purposes, these things are just easier to model as unidirectional changes getting worse. And it's really just about how fast you're changing in that direction. Akshat, I think we have a hand.
Hi.
Yeah.
Yes, I have a question. So, there was this very pretty famous research like recently that solved for aging in mice, right? Wherein they were able to reverse the markers that you're talking about within mice, and these mice were then able to uh, they seemed younger, right? So in that sense, if that is possible within mice, is that also possible within humans? And if so, like how does that sort of work within your research?
Yeah, I mean, I am. I followed a lot of that work. I'm not sure if I read that one in particular super closely, but I mean, I think in theory this is possible. At least it's possible to slow it down and maybe rejuvenate some of these things. Um, I think we've cured Alzheimer's disease like 12 times in mice. So the challenge here, and like that, I mean, I think we should keep trying to do that, and like we should keep trying to translate from mice to humans. The challenge is that it's really hard to translate from mice to humans, and uh there's large debates in the aging research field about this kind of thing because there's a camp that just basically says we're never going to do it. It's a waste of time. Stop trying to cure aging in mice. Like I'm not that extreme. I think that we can, but I think we should really focus on funding and encouraging not like translation. Like we have to, you have like if you're going to cure aging in mice, you got to do the next step to be like, how do you do this in a macaque? How do you kind of move up the chain of difficulty towards humans, or at least in wild type mice? So that's so many of these like cures in mice are in like, genetically inbred strains where every single animal is a perfect clone of another animal. And these are bizarre animals that couldn't survive like one day in the wild. So there's a lot of these translational problems that it's an important question, and I could kind of talk a lot about it, but it's fun to talk to. I have a lot of colleagues in the like mice and even like C. elegans worm aging world, and it's a very it's actually legitimately hard to talk to each other sometimes because of like how different the problem is in humans and mice. But I'm excited by the progress we've made in other animals. It at least to me suggests that we have it's worth keep working hard on humans because it's possible to have a complex biological system that you cure a really challenging disease like Alzheimer's. Okay, so I'm going to keep moving ahead, but feel free to jump in. So first off, I just want to kind of nail this a little bit better. What is an aging clock? So on the left-hand side here, I just have like a figure from a paper that just shows how a lot of clocks are trained. So what you do here is you make a bunch of measurements in people from, in this case, like from the age of 20 years old to 100 years old. And for brain clocks, you just feed in some high dimensional data set, like every single brain measure that you can get from a structural MRI scan. And you could toss in DTI, you could toss in, fMRI as well. And you're just training the machine learning model to predict how old someone is from those large high dimensional set of inputs. So this would be like what we call brain age. So you can see here in this paper, they did a really good job, right? Like from a high dimensional set of brain imaging metrics, you can get a correlation of 0.96 with age with a mean squared error of 3.8. So the average prediction is within 3.8 years of how old someone actually is. And that's pretty good. It's pretty impressive that you can do this. But ultimately, we don't want to know how old someone is because you could just ask them how old they are. We want to know how biologically old they are. So what they do is they take the difference between someone's prediction and someone's actual age. And in this case, we have someone whose brain looks like they're like 70 years old, even though their actual age is only 45. So that person seems to be aging faster because they have an older looking brain than their actual age. I call these clocks, these are often called first generation clocks. And there's really famous versions of these from epigenetics. You can take a blood draw, measure epigenetic changes. And these are called the Hannum and Horvath clocks. You do the exact, all of these are pretty much the same process. Just predict how old someone is and measure the difference between their actual age and the prediction. In MRI, if you use MRI to make the prediction, it's called the brain age gap. There's a paper that just came out in, I think it was Nature, and then a follow-up in Nature Medicine using proteomics to do the same thing, where you can make like organ-specific proteomic clocks, which is really cool work. But ultimately, underneath the hood, all you're doing is predicting someone's age and then subtracting to get like a gap. The second generation of these clocks are, instead of predicting someone's age, you predict their health or mortality risk. And these kind of these perform better in some cases than these first generation clocks. And I want to just highlight a couple challenges with these kind of first and to some extent second generation clocks. The first question is, what happens if we, in our field, a lot of like computer science type people have come in, and what they do is they make better and better brain age clocks. So they get excited about using different algorithms, doing all this fancy machine learning stuff, which is really cool. But what happens if you get like a perfect or really, 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 really good prediction of version of this clock? I'll just kind of say it, but ultimately what happens is you just get someone's age. And I hinted at this earlier, but like this is a problem because the better and better we get at training our machine learning models, the more we just converge to literally asking someone, what is your birthday? And that's not what we want to measure. And if it's a kind of fundamental problem, and I think anyone who works in AI stuff would agree with this, that you want to train a model to predict the signal of interest. And ultimately, brain age gap is noise, right? The difference between how old you are and how much the model predicted is noise. So we actually care about the noise. The signal that we are interested in is stuck in the noise of the model, or the error of the model. So that's a problem that I think we want to try to solve. A few other problems is that these kind of clocks, both first and second generation, are fundamentally cross-sectional by design. And this kind of is captured by my, I think a little nice short way of saying this is age is not aging. And this is key from like, I love this graph just because I'm I'm a nerdy history person. So if you think about like someone who is, let's just say someone who is born in 1940. So they're like 85 today, this would be someone maybe like our grandparents or great grandparents if they're still alive. So they were born like during or right after the Great Depression. They lived through, if you were in Europe or the US, you lived through World War II. Like a freezer, like putting your food in a freezer wasn't invented and like widespread until you were like 20 or 25 years old. And then like the expansion of like treatments for hypertension and cholesterol did not come until you were already like in midlife or beyond. So you can imagine that this person, like the exposures that they had throughout their life is just very different than the exposures that we all had in our lives because we didn't live through some of those things. A lot of these technologies were all already invented. And this is true, so if you're ultimately trying to predict someone's age and you just have a cross-sectional lineup of people from the ages of 10 to 100, this is a problem because partially what your model is predicting are just differences in exposure and what we might call cohort effects. And cohort effects are not aging, right? There's also the problem of survivorship bias. Anytime you line up a group of people from 10 to 100, the 100-year-olds are going to be really exceptional, right? Because most people die before they're 100. So you have this problem of like the 100-year-olds in your sample are actually kind of selected for especially good health. Another problem that we hit on is that the signal in those brain age clocks is in the air. We talked about that. So ultimately, like one solution to this is we actually want to predict aging. If we want to like build the best clock possible, we want that clock to predict the signal that we care about. And in this case, I'm going to argue that the signal we care about for aging is the progressive decline that involves multiple organ systems. So it's this longitudinal process of decline. And this is what is called the geroscience definition of aging. So within the geroscience field, this is how we define aging. So I want to tell you about my efforts to actually realize this. So I spent a lot of my grad school working with a study called the Dunedin study, which is, I talked about this briefly early in those facial photographs, but it's 1,000 people who were born in a single hospital on the South Island of New Zealand. And what's exceptional about this study is that they've done a really good job of keeping these people involved in research. So these people are now 52 years old, and at the last time we measured them, which is actually ongoing, But at the last full cohort, is at age 45, we had like 90, I think it was 94% of the original cohort was still in the study. So the real advantage of this is we have really in-depth phenotyping of an entire cohort that represents a really large range of socioeconomic and all kinds of other health outcome variables, because we didn't lose the people who like are typically lost in a lot of health research. So what we did is that over the course of about 20 years, we tracked biological deterioration in 19 biomarkers of a bunch of different organ systems. And I've listed them here on this little skeleton person. So we measured everything from how fast someone's how people's gums are receding, how many cavities they have, measures of like inflammation, measures of blood pressure and cardiorespiratory fitness, cholesterol, a bunch of kind of blood markers that you might get at your doctor that tell you about liver function and kidney function. So our goal was to measure just kind of like tried and true gold standard measures of health across the body, across a bunch of different organ systems, and track it over the course of 20 years. And importantly, this is happening in kind of like early and midlife. So this does not reflect, like our measurements here do not reflect age-related disease. They really reflect kind of changes across the lifespan in people who have, who are relatively healthy, but are aging, or these biomarkers are changing at different rates. So we have 19 of these biomarkers, and we measured them across 20 years. And what we're going to do is take a pretty kind of basic approach that I think is pretty powerful. And we're just going to model how fast each one of these biomarkers is changing. So it's end up being 19 biomarkers. And we're going to z-score all those slopes, z-score all these values, generate slopes for every single individual, and then just take the average slope. And then we're going to scale that so that the average rate of aging in this is 1. So what it means is that someone aged 1 biological year per chronological year. And then what you can do is put everyone on this distribution and see how big the individual differences are in the rate of biological aging, which is what we're ultimately trying to get at. And this is across organ systems, right? So it's not any one organ system. This is kind of the average of all of the organ systems. And this gets at one of the questions that someone brought up earlier, is that we could debate whether or not this is the best way to do this. But the goal here is to measure the overall rate of change. You can imagine creating like a pace of aging clock for a single organ, right? Maybe we really just care about the heart, or maybe we really just care about the liver or something like that. In this case, we're taking an approach to really try to measure the kind of full trajectory of aging and get this average measure across biomarkers. And there's some pros and cons to either approach. But what you can see here is that the average person is aging at a rate of aging of 1. So 1 biological year per chronological year. But there are people, even at the age of 45, who seem to be aging about twice as fast or about half as fast. So we see like pretty broad individual differences between the ages of 26 and 45. So what we're going to do to try to make a better brain aging clock is I trained a model to predict this measure. And so we're predicting how fast someone is aging in this cohort using MRI scans at age 45 to say, how fast did you biologically age over the last 20 years? And I'm just going to feed it in a big high dimensional set of free surfer measurements. So these are kind of like run-of-the-mill things you can get, just run recon all on a structural T1 scan. Importantly, we trained this model to predict the pace of aging from measurements that you can get from a single 6-minute MRI scan. So this is relatively accessible. Almost every single MRI study is going to collect a structural MRI study. So you can estimate our clock from any MRI data set out there. And we're going to predict how fast someone is aging using this pace of aging measure. And we're going to just use cross-validation to train these weights. And what we get is that we can do an okay job. This is not like perfect prediction, but it's pretty good from the fact that we're just using a single MRI scan to predict 20 years of biological change. And that's an important piece, is that what we're training is we're training a model to predict biological change from a cross-sectional measure. So fundamentally, this is going to be a proxy for longitudinal change. But we can measure individual differences, and we're at least capturing something here. So all of this that I've shown you so far was just within the Dunedin study. So it's just within those 1,000 individuals from the South Island of New Zealand. But if we have a clock that's actually we think works and we think measures something about biological aging, we need to be able to show that it generalizes, that you can apply this predictive model and apply it to a data set that is outside of the training sample. So that's what we did next, is that we took these weights and we applied them to the ADNI study, which is the Alzheimer's Disease Neuroimaging Initiative, which is about 1,700 scans. And this is a study of Alzheimer's disease and healthy aging. And what they did is they measured changes in cognitive status. So someone who developed mild cognitive impairment or Alzheimer's disease, dementia conversion. We measured just how fast their hippocampus is atrophying, and other features of cognition and education status. We also applied this model, so this was in the US primarily. We also applied this model to a big sample in the United Kingdom called the UK Biobank. I'm sure many of you have heard of this. It's a big kind of population study of, it's actually bigger than this now, but at the time we downloaded it, it was about 42,000 MRI scans. And they have all kinds of measures of health and cognition and outcomes of chronic disease and mortality. So we're taking a model that was trained on a single sample of 45-year-olds in New Zealand, and we're asking, if you apply this model to new scanners in other continents, how well can we predict something that actually is measuring biological aging? So we did a handful of studies to try to validate like whether or not this is working. So first off, having a faster DunedinPACNI or a faster rate of aging is predictive of lower cognitive performance. And I just have a bunch of effect sizes laid out here in ADNI on the left and the UK Biobank on the right. And we just went through like common cognitive assessments that are often used in aging outcomes. So things like fluid IQ, working memory, trails, matrix reasoning, digit symbol substitution. And you can see that we can our measure seems to be tracking individual differences in cognition. So that's like a start. This is just cross-sectional. People with a faster DunedinPACNI were more likely to have cognitive impairment. This is in the ADNI study. So the people who seem to have the faster rates of aging were more likely to have dementia. And it's kind of the stepwise, then MCI, then cognitively normal. And importantly, both of those things I just showed you was all cross-sectional. It was just saying, if your brain looks like this, were you more likely to have dementia, and were you more likely to have or perform worse on cognitive tests? But what we really care about, if we care about aging, are longitudinal outcomes, right? We want to be able to say that, like, if we think you're aging faster, I can predict something about what's going to happen in the future to you, right? Like, if you're aging faster, in this case, I would expect you to develop cognitive impairment more often than someone who's aging slower. So that's what we tested here. Individuals who are cognitively normal at baseline, followed up to 8 years later, were more likely to develop a diagnosis of dementia. And that's kind of the survival curves I'm showing on the left. And on the right, I'm showing people who had mild cognitive impairment and went on to receive a diagnosis of dementia at follow-up. And you can see in both these cases, we have like pretty decent sized hazard ratios that people who seem to be aging faster based on how their brain looks were more likely to develop diagnosis as we followed them up. And this is also true of chronic disease. If we follow, we just categorize a bunch of age-related diseases, and we asked if you've never had this disease, how likely are you to develop this disease at follow-up? People with a faster rate of aging or an older-looking brain were more likely to develop chronic disease. They were also more likely to die. The UK Biobank has a really great registry of death records. And you can see that people who had older looking brains were more likely to die in the next 7 years. And importantly, just to kind of wrap this up, is like I'm trying to make the argument that our measure is capturing something that's different or that's better than these previous brain age gap measures. Importantly, in both ADNI and UK Biobank, PACNI is loosely or kind of weakly correlated with brain age gap. So they're measuring something that's similar, but they're definitely measuring a lot of kind of new information. So does our measure outperform brain age gap in predicting aging-related outcomes? That's kind of the final question. And in this figure, I just plotted a bunch of effect sizes. And a lot of these are the effect sizes you saw in the previous results. And in blue, we have our PACNI measure. So this is how strong is the association between PACNI and, in this case, a measure of cognitive functioning, memory, psychomotor speed. And in each case, we have the blue effect size is PACNI, and the pink effect size is that first generation brain age gap measure. And what you can see is that in almost every case, our new measure, this PACNI score, is at least as good as the brain age gap, and in many cases, it is better. And this is especially true in the UK Biobank, and it seems to be especially true of measures like health-related outcomes and kind of longitudinal changes, like how fast your hippocampus is going to decline in the future. So this is all kind of a big whirlwind of results. It summarizes our first steps in validating this PACNI measure. There's a bunch of new research that's ongoing in kind of building on this. But just to conclude, we think that structural neuroimaging can be used to estimate individual differences in the longitudinal rate of biological aging. Faster DunedinPACNI is associated with poor cognition, cognitive decline, dementia risk, and mortality. And we at least have initial evidence to say that DunedinPACNI is distinct from those first generation brain clocks like brain age gap. And just to make the plug quick before I end up and open up for questions, is that we have a GitHub page. It should be easy to find from our paper. I'm happy to send that to anybody too. So if you have an MRI data set that you're working with and you want to apply generate our PACNI measure, it's super easy. You just process it with FreeSurfer and upload the data, and it's pretty quick and easy. And then I just want to say thank you. Here's a bunch of the people who are like really important in that Dunedin study on the left-hand side, our funding and the other kind of publicly available data sets that we used. And this paper is now out in Nature Aging. So take a look at it if you want to learn more. But happy to have a discussion and have a little bit of a chat as well.
So it's pretty nice. Any questions? I think there's one in the chat. What if some biomarkers age at a very high rate, but others age slowly? So how do we deal with this scenario?
Yeah, so the kind of nice thing about our approach is what we care about is really just individual differences. So kind of like what would it mean for one biomarker to age fast and another one to age slow is kind of like, I think it doesn't really matter for our way of looking at this because the kind of units are just whatever that measure is. So it would be like, how fast are your gums receding? Or how fast is your cholesterol going up? So what we are not paying any attention really to these means. What we're trying to quantify is variation around the mean. So anything that all of those 19 biomarkers that we used, we specifically selected them because they showed large amounts of variation within our age window of 26 to 45. Because you can imagine a biomarker where like everyone declines at the same rate. So there's a huge age effect, but there's almost no differences between people. And that would be kind of worthless for our purpose. Um, there's actually, I feel like there's good examples of this that I'm not thinking of off the top of my head. But um, what we really want is measures that seem to change with age, but show large individual differences. And that's what we're kind of selecting for in our biomarker selection process.
So like when
It's
you're
a good question.
yeah, so when you're selecting these biomarkers, you're looking at that individual difference as the age. So is there like a standardized method you're trying to get at, or?
Yeah, so the way we selected biomarkers, it was multiple steps. I'm kind of just jumping through some of them. What we did was we wanted like tried and true measures that like are commonly used and are have well-established relationships with age-related disease. Um, and then importantly, like our sample, and I think there's good reasons, and I can get into it, for why we were really interested in biological aging in kind of the first half of the lifespan. So between 26 and 45. So we only cared about biomarkers that like made sense to measure in those people. So we kind of sub-selected measures for that. And then we had to have measures on them over the course of 20 years in our sample. So they weren't necessarily the best, but I think they're actually a lot of the measures that people would commonly use. And so that was kind of multiple steps in us kind of filtering down to these 19 biomarkers. But ultimately, our goal was just to quantify like variation between people in aging rates in biomarkers of kind of organ function. Because that's kind of what the geroscience definition of aging is really getting at, is that it's a correlated decline. It's not a single, if you just have like your liver function just dropping off a cliff, it means you probably have liver disease. It doesn't mean that you're aging quickly.
Yeah.
And what we really wanted to capture was this like common trajectory that's across multiple biomarkers.
Any other questions?
Looks like there's a few in the chat.
I think Prakriti here is asking, thank you for the talk. What are your thoughts on individuals like Brian Johnson who are making an attempt at reversing their bioage through extreme measures like exposure to pressurized oxygen tanks, sound wave therapy, etc.? Is there any substance to this method? Any thoughts?
Yes. Yeah. I mean, I've followed a Brian Johnson a little bit. Not, I wouldn't say that I've gone fully into his stuff. I think he's an interesting character. I think I mean, I'm a big fan of like, everybody try their own thing, and he's really taking an extreme approach to trying to die. If people haven't heard of him, this is a guy who like he's really trying to like slow his personal aging rate down as much as possible. And he's like, fully in literature, and he's taking all these supplements and he's doing all these therapies, and anything he can convince himself is scientifically justified, he's going to do it to himself and see if he can like extend his lifespan. And he's like an extreme version of this. Like this is his full-time gig. It's just doing this. And I I can't help. I have to say it. There was this hilarious, I think, Twitter conversation where there's a paper that came out recently that showed this like really kind of elegant biology paper where if you've ever seen these, they compare across like every single species they can find to see if there's some like biologically conserved path to age-related changes. So this one was comparing across, I think it was like, 40 species or something like that. And what they showed is that there's this really conserved mechanism that if you have males of a species and you castrate them, that they live longer. So if you cut off their genitals, they live longer. And there is this whole nice, funny little Twitter feud about saying, here you go, Brian, like give it a try. Like if you're gonna do everything, are you fully committed? And I thought it was it was a funny, poke at how far are you willing to go for this age-related flowing. So I honestly, I would say like these kinds of specific interventions, I don't have strong opinions on. I'm more kind of interested in these basic processes. And if there is a really good intervention that we have good evidence for, I want to be able to give someone a tool to actually test if it's working.
The one more in the chat is was the gut microbiota also considered for the finalized biomarker selection?
We've talked about gut microbiome stuff, and I think there's some really cool stuff. We didn't have good data in this cohort because anything that made it into this cohort, we had to have measured longitudinally over the course of 20 years. So this is going back to like the late 90s. It had to be in our protocol. And I think the microbiome stuff has really accelerated in the last like 10 years or so. So we didn't have good measures going back. But I think it would be, I think it's being either we might have some measures of it now in people's 40s and 50s. So it could make it in eventually if we follow this group long enough.
Nice. One question, what could be the ideal pace of aging in the 26 to 45 years? Does it have to be 1 or a value of 0.85 is also considered good? How can we differentiate between healthy aging and aging due to, let's say, Parkinson's or Alzheimer's?
Yeah. No, I mean, these are like core questions that I think the field is really kind of grappling with and thinking about. Something that I'm very interested in, and I have some collaborators who work on this, is like what people are commonly calling like super aging. So people who are exceptional agers, and you can measure this in different ways, but I'm fascinated by this process of like, first off, like, is it a real thing? Like intuitively, we all think it's a real thing, right? Like we know someone who's like 70 and they look like they're 40, and it's just like, what's going on there? But it's harder to measure and it's harder to think through what it would mean than it seems at first pass. In our measures, like in our 19 biomarkers that we measured across 20 years, the best case scenario would really be to have the slowest. So it would be less than 1. It would be like 0.5, I think, was the lowest person in our measure. And what that means is that you've barely changed in terms of your cholesterol levels and heart functioning from the age of 26 to 45. And you've those things have gotten worse at half the rate of someone who is the average ager. So in most of this kind of aging stuff, like after you're the age of 25 or so, it's really good to just hold on to these biological things as long as you can, which is kind of sad to say, but it's just the way it is that most of us, like our livers were functioning at their peaks when we were 25. And that is, it's kind of a slow, steady decline from there. And most of us don't lose that much between the age of 25 and 40 or 50. And there's definitely these like non-linear changes once you get older where things accelerate more quickly. But holding on and really slowing down that decline as much as possible is probably the best case scenario. And the other question was like Parkinson's. Yeah, I mean, I think that's a really key distinction is like once you start developing age-related diseases like Parkinson's, like Alzheimer's, like, even a lot of cancers are age-related. What's the difference between normal aging and a disease state? I think is a really fascinating and big question that I think about a lot, and I don't think we have great answers for. There's some clear cases where like a disease state is clearly not aging. But there's a lot of more edge cases where there's a big gray area between what we'd call disease and what we'd call normal aging. And I think it's a really cool area of research that people continue to think about.
I think there's one more question on like what's next after this? What is the direction you want to take?
Yeah. I mean, I think there's just a ton. We have several questions that are just really just like further validating this metric. Just finding new big data sets to generate PACNI scores in, and ask questions about aging, and ask questions about what are risk factors for aging, and what seems to accelerate or slow down aging. And the real, like, I'm very focused on getting deeper into the problem of like, ultimately what this is a proxy. So you're trying to predict how fast you're aging from a brain scan, and how good it is an open question, and how useful it is. Like, could you really run a trial where you're doing an RCT of some intervention, like maybe some supplement you take that we think slows down aging? Would this measure be a good outcome measure? And to get there is a lot of hard work that we're not very close to. But I think digging deeper into really thinking about what longitudinal changes look like within individuals and how sensitive it is to interventions is something I'm very interested in. But also, I mean, we made it publicly available so that hopefully everyone, like tons of researchers, can jump in and ask some of these questions too. So if you're interested and you want a project, think about it. Go to the GitHub page.
So they are the class people have a project where they're analyzing open neuro data set, running FreeSurfer, fMRI on it. So if someone wants to take it up, they can feel free. Again, if you have questions, you can reach out to Max. There's one more question on if we have to. So can these biomarkers be used for infants and young children as well? Are they similar, or again, is it different from maturing and development, or is it like between the age of 26 and 45 where we can use this?
Yeah. I think it's like it's a question that a lot of people have had of like, what's the differences between development and aging? And I would be very careful in applying my measure to anyone below the age of like 18, probably. We've messed around a little bit in some data sets that cover younger people, but I think it's complicated and it's messy, and it probably doesn't make sense to interpret like biological aging in like young kids. I think there might be a different way to measure it, but our tool is probably not the best way to do it.
Rajat, I'm a bit confused about your question. Can you ask that? Yes.
Yeah, I was asking when you are applying this technique. So, are you taking the every possible info that you can get from a scan and then you are comparing it with the same individual, or is it limited to some specific parameters that you are looking in a particular scan?
Like, what are the parameters from a scan that we care about? Is that kind of the question?
Yeah. Or is it the full scan, like every possible info that you can get from a single scan?
Oh, yes. Yeah. I mean, so far, the prediction is, I mean, the goal is to make this widely accessible and easy to apply, because we thought we could do a pretty decent job even with kind of widely available metrics. So what we're using is like I mean, it sounds like you all are maybe running FreeSurfer. So it's the outputs of FreeSurfer. So it's like ASEG and APARC, which is like cortical thickness and surface area and subcortical volumes. And then we include gray-white ratio, which is like a metric that FreeSurfer generates, but you just have to know where to look for it. So we started there because our goal was to like, can we make something that could actually be used in research in most labs? And it works pretty well. But I think there's a lot of room for improvement. So a lot of people in like the brain age literature have tried to use voxel-wise metrics and try to do a lot of fancy, extract, millions and millions and millions of parameters. I think I'd be very open to anyone who thinks they have a good tool for doing that. The problem is that the more fancy you make it, potentially the harder it is for other people to actually use it. But that's kind of how that's the approach we've taken so far.
Thank you, Max.
Yeah.
Cool. So I think we can take one more question before we close. Anyone? Okay. Cool. Awesome. So let's give a big round of applause to Max again. Thank you. And if you people have questions, can they write to you?
Yeah, I'd be happy to email with anybody who asks questions. But it was really nice to meet you all, and don't give Vaibhav a little bit of a hard time, just only not too much, but he needs a little pokes. It's nice to see you, Vaibhav, and best of luck.